For water use efficiency, there's been a couple of examples, in particular from ICRISAT, where drought-tolerant genotypes start out the season by taking up water at a lower rate, and then by the end of the study, during grain filling, they have higher water uptake rates.
Yeah.
So, in principle, from what you're saying, they shouldn't be successful because they're leaving extra water for other genotypes to take up.
I wouldn't expect that to evolve under natural selection unless the plants somehow have had a monopoly on some volume of soil.
But don't you think we have to keep in mind that water uptake is not uniform within a soil profile? Like, the plants aren't just leaving all of their water right at the top. Maybe it's at different depths where they're extracting water or not extracting water, and
Sure.
maybe the weeds can't take it up if they have to grow through dry soil to be able to get there.
Yeah, that's certainly a possibility, but we have to realize that natural selection is driven first and foremost by competition within a species. So presumably over the history of these plants, it's common to have, two sorghum plants near each other. And I mean, deeper water, if you look at a typical rooting pattern, it's probably easier to steal surface water from your neighbor than it is deeper water. So, I think, you've got to, I certainly agree with your point that we need to think about this. But I don't think I would say that natural selection would necessarily favor leaving deeper water in a competitive environment where that deeper water might be taken by somebody else. But we can do it. We can select for it. I'm meeting with you today, so we'll talk about this more. Yes?
I didn't quite understand. The cartoon you showed just before this slide.
Thank you for asking. Go ahead.
And here, your hypothesis is that beneficial rhizobium from soybean is going to maize, but maize does not enter in symbiosis. So, this could be very well not because of rhizobium, but something else.
Yes. If that wasn't clear, that's what I meant.
Okay.
That this could be an effect of the soybean on mycorrhizae, for example, or it could be an effect of the soybean on soil structure, but not
rhizobium.
Not rhizobium. This is rhizobium. This is very unlikely to be rhizobium.
I have another question.
Yes.
More like philosophical. Why, and you're talking about cheaters rhizobium. Why nature didn't solve this problem by having an obligate symbiosis, where rhizobium is always, throughout going through the reproduction cycle and they never separate?
Well, it did in a couple of cases. Mitochondria and chloroplasts. They solved, those were originally symbionts, and the problem of cheaters was solved by linking the reproduction of the mitochondria and the chloroplasts inextricably to their host cells. Why that hasn't happened in the case of the rhizobia, maybe we need to wait another 100 million years, maybe a complicated sort of thing. The evolutionary theory of cooperation between species is pretty clear. There are two things that ought to favor cooperation between two species. One is if the symbiont is transmitted in the seeds, then, if the plant dies before it makes seeds, you have no transmission. So that ought to select for more beneficial, more cooperative symbionts. But even in that case, if you have two genotypes in the same host, then you have this sort of a tragedy of the commons situation where, it's best for both of them if the host survives, but You do the work to make the host survive. I'm going to go and get the seeds. Okay, so there's that kind of a conflict of interest among symbionts within a plant that could undermine cooperation, even in the case of a seed transmitted or vertically transmitted, as we call it, symbiont. So that's the theory. Now, here's a couple real-world examples. Soybeans, or legumes and rhizobia in general, the seeds, the rhizobia are not transmitted through the seeds. They're transmitted horizontally. It's fine with the rhizobia if the plant dies. It doesn't affect their ability to find the new host. And you always have multiple, maybe 10 different genotypes per plant. So it seems like, according to theory, it should evolve into parasites. And the reason it hasn't, I'm pretty sure, is because of these host sanctions that we've described. So that makes me then, if you think about other kinds of endophytes where you don't have those sanctions, I'm not optimistic. But here's another example. There are these endophytic fungi in wild grasses where you have a single genotype usually per plant and where they're transmitted through seeds, and many of them are not beneficial to their host.
I'll give you a classic example actually. There is one symbiosis where it's obligate symbiosis, NRB-Nazuli.
Yes.
It's very productive. It's
yes.
not what you mentioned, the fungal symbiosis. So there is an example there.
Yes. Yeah, well, certainly rhizobia are more beneficial than not. Mycorrhizae are more beneficial than not, and I wondered about that because it seemed impossible to me that plants could impose sanctions on mycorrhizae because you're going to have two different genotypes within 10 microns of each other. How can they possibly discriminate between them? But Toby Keers, the same woman that did this soybean work, is now a professor at Amsterdam, and last year she showed that in fact plants do impose sanctions on mycorrhizae that provide less phosphorus. We don't know the mechanism yet. But yes, if I had nothing better to do, I'd actually like to work on Isola. It's a very interesting system. Okay? Up there? Yes. Feel free to leave. Anybody needs to leave.
You commented on the fact it would be rather nice to. Increase photosynthesis.
Yes.
In these. And when I was in the seminar, I pointed out that when I was at Davis, one of the alfalfa plants we've been looking at in relation to photosynthesis and nitrogen fixation had a rate of photosynthesis viewed to be very equivalent to maize.
Yeah.
The problem was that was a space plant. When they were put into a community, the rate of photosynthesis fell because the specific area started to change.
Yeah.
You got that competitive effect from, I guess, far-red ratio changes. I wonder whether, those kinds of sensitivities which control, I suppose, to some extent, competitive ability and are deeply rooted, could in fact be changed somewhat to improve photosynthesis, or whether they're too deeply layered in the psyche of the plants, as it were.
Yeah, I think that's a real interesting question, and I don't know the answer, but I would not assume that we can't make changes in things like how specific leaf weight responds to red, far red. I think we probably can make those, well, I don't know, but we might be able to make those changes, and we might be able to make changes that were rejected by natural selection but are nonetheless beneficial in agriculture. Incidentally, that paper with the Drysdale wheat has, I was referred to it as an example of trade-off-free improvements. And then you read the paper, and it's this, brilliant discussion of all the trade-offs that are involved in increasing water use efficiency in crops. And, so it's a wonderful paper in terms of a practical success, but it's also a very nice theoretical discussion of, well, if you have thicker leaves, you have higher photosynthesis per unit leaf area, but if you have a limited amount of resources, early in the season, making thicker leaves means not having as much leaf area, and that means you have more photons that are hitting the soil, evaporating water, not contributing to photosynthesis, and so on. So I discuss it in some detail in my book, but it's really a wonderful paper. You had a question?
My question is for the allelopathic effects, how that will play in the heat suppression.
Yeah, good question. I used to read a little bit about allelopathy, but I'm really out of date. Well, let me throw you out just a sort of an evolutionary perspective on allelopathy. There's some interesting stuff on allelopathy. Some cases where these desert shrubs were thought to be allelopathic because nothing grew around them, and then somebody figured out it was because the mice lived in the shrub and would go out and eat all the seeds that were, shed nearby. And an awful lot of the early allelopathy literature that I looked at was effect of allelopathic chemicals on germination of seeds. So think about this from an evolutionary perspective. Two alternatives. There's an allelopathic chemical that kills seeds. Well, that's going to strongly select for immunity or resistance to that toxin. And of course, the allelopathic plant is getting a much higher dose than the seeds are. So it seems like a difficult thing to kill seeds and not have them evolve resistance as they evolve resistance to herbicides and so on. But most of these studies, what they measured is lack of germination, not lack of viability. So now think about it from the seed's point of view. And here's this chemical that's being produced by these other big plants. Oh, there's big plants here. If I germinate, I'm coming up under a big plant. There's not going to be any water left on the soil surface. They're going to shade me out. This is not a good time to germinate. I should wait until these plants die, get grazed or killed by a forest fire or range fire or something like that. So now the question is, natural selection in response to these chemicals in the target seeds. It's the opposite direction. You're selecting for the ability to detect and respond to low concentrations of these chemical cues, these information-bearing molecules that tell you that there's a lot of competitors and it's not a good time to germinate. So given the improbability of seeds not evolving resistance to being killed, and the high probability of them evolving the ability to detect chemicals from other plants, I wonder whether some of the cases that we're calling allelopathy and seeing failure of seeds to germinate is actually almost a cooperation between the two species, and the seeds going, okay, I can germinate now. I can live for 10 years, I'll germinate the next chance to get. Just a thought. I should probably let people move on. Thanks for your great questions.